All rights reserved, we reserve all rights. The End of Germ Theory. The nearly 460-year-old idea that germs, and more recently theoretical viruses, cause illness, disease, and death is known as the germ theory of disease. The reason it cannot be called germ fact to this day is because there are two very simple ways to prove it. Both have been done, and not only could they not prove it, but both disproved it. This is not to say that there are not other bioelectric mechanisms, bioresonant communication, and shared mental, pharmaceutical, and environmental factors that do make it seem that certain people do get the same detox symptoms we call disease or cellular dis-ease after being around each other, which we will cover at the end of this film. But as you will see, it is neither germs nor theoretical virus particles that are the cause of such phenomenon. The first, very simple way to prove germ contagion theory is to expose hundreds of healthy people in every way possible to the breath and body fluids of sick people and see if those healthy people get sick from the exposure, which was done during the 1918 alleged Spanish flu pandemic by Dr. Milton J. Rosenau, along with the U.S. Navy and U.S. Public Health Service. The volunteers were all of the most susceptible age, mostly between 18 and 25 years old, only a few of them around 30 years old, and all were in good physical condition. None of these volunteers, 100 in all, had influenza. They proceeded rather cautiously at first by administering what they claimed was a pure culture of bacillus of influenza, meaning bacteria found in the boogers and lung fluid of people with Spanish flu symptoms, called Pfeiffer's bacillus, in a rather moderate or small amount, sprayed into the nostrils of a few of the healthy volunteers. But all of those trials proved negative, meaning none of those volunteers who got bacteria from people sick with the Spanish flu sprayed up into their nostrils got sick with the flu themselves. So they became bolder and selected 19 of their 100 volunteers and gave each one of them a very large quantity of a mixture of 13 different strains of the Pfeiffer bacillus that they had recently obtained from the lungs at necropsy, meaning lung fluid from a dead corpse who died of pneumonia. These 13 corpse-retrieved organisms were sprayed in an atomizer into the nose and the eyes of the healthy volunteers and back into their throats, while the healthy volunteers were breathing in. They used some billions of these bacterial organisms according to their own estimated counts, but none of the healthy volunteers got sick. Next, they collected the snot and mucus secretions from the mouth, nose, throat, and lung bronchi from the sick people and transferred this mixture into their healthy volunteers. The snot was blown from the sick nostrils into a tray, bronchial mucus was obtained by coughing up the lung phlegm, then swabs were taken from the mucus surface of the nostrils and also the mucous membrane of the throat. They mixed all that up together and sprayed it into each nostril and into the throat of the healthy volunteer while they inhaled, and sprayed it on their eyes. None of these people got sick. Some of that same mixture was then filtered and injected into other volunteers, but produced no results. None of them got sick. Next, they took new snot and phlegm samples from new sick people. This time, they sprayed a much larger amount into their nostrils, throat, and eyes, and then they made the healthy volunteers swallow the rest of the sick mucus, but none of them got sick. Next, they tried immediate direct transfer from sick person to healthy person, taking little cotton swabs on the end of sticks and transferred the sick mucus directly from nose to nose and throat to throat to the 19 healthy volunteers. And they used a special throat tube for the throat culture to make sure they got not only the material from the tonsils, but also from the posterior nasopharynx, way in the back of the throat. None of the healthy volunteers who received the material directly transferred from sick cases got sick themselves in any way. And this procedure was done to all 19 volunteers at least twice, and some of them 3 times, but none of them got sick. So next they tried an experiment using injections of blood. Blood from the sick people were injected into each of 10 new healthy volunteers, but none of them got sick in any way. So they collected a lot of mucus material from the upper respiratory tract of sick people, ran that through a filter, and injected that phlegm into 10 new volunteers with a needle, and none of them got sick in any way. Puzzled and frustrated, next they led a healthy volunteer to the bedside of a sick patient. He sat down alongside the bed of the patient. They shook hands, got as close as he conveniently could, and they talked for 5 minutes. At the end of the 5 minutes, the sick person breathed out as hard as he could, while the volunteer's face was muzzle to muzzle, 2 inches apart from the sick breath, and was instructed to breathe in as the other breathed out in his mouth. They repeated this 5 times in a row, and after that, the sick patient coughed directly into the face of the healthy volunteer 5 different times. Then after all of that, they moved the healthy volunteer to the next sick patient selected and repeated that entire procedure until each of the 10 healthy volunteers had done this with 10 sick people in a row. Then they were all watched carefully for 7 days, and you guessed it, none of them got sick. Not one. They tried another set of experiments with 50 people that also failed, that he said was too long to go into, and they tried another one with 6 people that failed, and then they gave up. Not only did they not prove the Spanish flu was contagious, but they clearly disproved it. And Dr. Rosenau wasn't even the first to fail, because at the end of his paper he cites that he already knew that Dr. McCoy and Dr. Ritchie did a similar set of experiments on Goat Island Quarantine Station off San Francisco, and used volunteers who had not been exposed to the Spanish flu outbreak at all, and they too had negative results. They were unable to reproduce the disease. Sure enough, we see it in the public health reports published January 10th, 1919, called Some Interesting Though Unsuccessful Attempts to Transmit Spanish Influenza Experimentally. The San Francisco Angel or Goat Island Quarantine Station experiments, carried out by Dr. G.W. McCoy of the United States Public Health Service and Lieutenant Dwayne Ritchie of the United States Navy. A group of 10 men were divided into 2 squads of 5. One group had put up their nostrils a heavy suspension of liquid cultures made of 8 strains of Pfeiffer's bacillus bacteria without filtration. The other group had the same mixture but put through a Berkefield candle purifier. The results were negative. Nobody got sick after being observed for 7 days. Next, 40 volunteers were put in 4 groups of 10 each, and liquid secretions of phlegm and mucus from the upper respiratory passages of people sick with influenza within the last 1 or 2 days of onset were put up their noses with both medicine droppers and sprayed up their nose with atomizers. In every case, the results were negative. So far as the reproduction of influenza is concerned, nobody got sick. Next, they made a filtered liquid from the phlegm and mucus taken from the upper air passages of a person sick with acute influenza, and used a dropper to put it in the mucous membrane of the eyes of 2 volunteers. And then the same phlegm and mucus was injected into the skin of a 3rd volunteer. In each case, the results were negative. None of them got sick. So finally, just like Dr. Rosenau, They took blood from a person sick with influenza and injected it into one healthy volunteer with negative results. He did not get sick. How could the Spanish flu have been some highly contagious disease if teams of medical doctors and volunteers bending over backwards and doing cartwheels couldn't even make just one person sick after hundreds of volunteers and attempts? Short answer, it couldn't. Around this same time, in October 1919, in the Journal of Medicine, we read a letter to the editor from a New York doctor, Alfred F. Hess, titled, "Need of Further Research on the Transmissibility of Measles and Varicella, which is chickenpox." Dr. Hess wrote to the editor, "I have just read the abstract in the journal of Dr. Sellard's article on the insusceptibility of man to inoculation with blood from measles patients, carried out at Johns Hopkins, September 1919. It is remarkable that Dr. Sellards was unable to produce this highly infectious disease by means of the blood or the nasal secretion of infected individuals into healthy volunteers. Not long ago, however, I had a similar experience with varicella, meaning chickenpox. Thus we are confronted with two diseases, the two most infectious diseases of the endemic diseases in this part of the world, which we are unable to transmit artificially from man to man." It is remarkable that in the middle of the Spanish flu pandemic, he doesn't mention the Spanish flu as one of the two most infectious endemic diseases in that part of the world at that time. But more remarkable that here again, we have two more doctors who failed to produce contagious transmission with two other alleged contagious germ diseases. As we fast forward from the 1918 Spanish flu pandemic to the polio contagion experiments carried out in 1949 and the early 1950s, we run into the exact same problem. On page 105 of her 1957 book, The Poison Needle, Dr. Eleanor McBean tells us, "During one of our most widespread polio epidemics in 1949, contraction of polio or catching polio by definite contact with other victims of the polio disease was not established in an elaborate study made by the New York State Health Department. The United States Public Health Service in its studies also found the same negative answer during succeeding outbreaks of polio later. In short, they learned that the disease was not contagious." Time magazine commented on this unexpected revelation of these surveys by remarking that when and where people catch polio remains a mystery. The supposition, meaning an assumption based on zero fact or evidence, that viruses entered through the mouth and nose led to the experimenters to make exhaustive tests on both animals and human beings, mostly prisoners and orphans in institutions. They were exposed by close association or close contact to polio patients, and their throats and nasal passages were frequently swabbed with matter from the sick patients that was supposed to contain viruses. No polio was produced in this way, and about the only noticeable effect was that the subjects of the experiments lost their sense of taste and smell due to the damaging effect of the poisons in the serum that was used on the test swabs. And we're going to examine one of these failed polio studies together later in this film so you can see for yourself. Johns Hopkins magazine tells us, into the 1950s and in some cases longer, physicians had experimented on prisoners, orphans, soldiers, and even newborn infants without the full consent of the subjects or by deceiving the subjects about the purpose of intervention. But deceiving the public about viruses and vaccines is where they morally draw the line. But think about it. If some government food agency or sandwich shop kept assuring you that this was chicken noodle soup, but kept serving you this sandwich instead, at what point would you start to question the legitimacy of that claim? In order to understand the logical fallacies in the thinking of big pharma-sponsored virologists and see the obvious flaws in the petri dish experiments they use as proof that their unproven germ theory is possibly correct, we need to go back in time again and ask, what is polio, and what caused the Spanish flu, if not germs? Dr. Eleanor McBean was actually there, as were her two doctor parents and colleagues who were healing the victims. According to them, ironically, the Spanish flu pandemic was caused by multiple toxic vaccines, kept alive with the addition of more poisonous drugs administered by the doctors who tried to suppress the symptoms caused by the vaccines themselves. We know that the Rockefeller Institute for Medical Research carried out a large-scale bacterial meningitis vaccination campaign in the U.S. Army in 1917 and 1918, supervised by Frederick Lamont Gates, under supervision of his father, Frederick Taylor Gates. We can see one of the published studies carried out on soldiers in Fort Riley, Kansas, in late 1917, the start of the Spanish flu pandemic, where soldiers were being given 3 increasing dosage amounts of bacterial meningitis vaccines in 4 to 10-day intervals. Even if you are ignorantly pro-vax, would you go out and take 3 increasing doses of polio, flu shots, or COVID-19 vaccinations in 2 weeks? They called it the Spanish flu, yet Spain remained neutral in World War I, and German submarines patrolled their coast, so the U.S. didn't even have soldiers and bases in Spain. It also took 6 to 8 days just to sail troops home from France to the U.S., never mind the extra time to get home to the other states and bases. Yet, the first reported Spanish flu cases all broke out in the exact same time, not in Europe, but all around the United States, shown here in green and yellow, even in Southern California and, of course, Fort Riley, Kansas. When you lay the map of important U.S. military bases where mass vaccinations were taking place over the outbreak map, they match exactly. Dr. McBean's research into military vaccination records uncovered that they were not only giving these soldiers bacterial meningitis vaccines. That was only one of the 14 to 25 different shots given to the soldiers. As we ourselves have just seen, after a COVID-19 vaccine safety data document that they had hoped to remain sealed for 75 years was published, showing on page 30 a 9-page list of over 1,300 diseases already caused by just one COVID-19 vaccine worldwide, and that's just from one manufacturer. So what kind of diseases were these soldiers getting from a combination of 14 to 25 different vaccines at once? Many of the soldiers were insane from post-vaccinal encephalitis, but the doctors called it shell shock, even though many of the soldiers had never left American soil. The conglomerate disease brought on by the 14 to 25 poisonous vaccines baffled the doctors, as they had never had a vaccination spree before, which used so many different vaccines. The vaccines brought on high fever, extreme weakness, abdominal rash, and intestinal disturbances, characteristic of typhoid. The diphtheria vaccine caused lung congestion, chills and fever, swollen sore throat, clogged with the false membrane, and the choking suffocation because of difficulty in breathing, followed by gasping and death, after which the body turned black from stagnant blood that had been deprived of oxygen in the suffocation stages. In the early days, they called it Black Death. Does this look like any flu symptoms you've ever experienced or seen in your life? The other vaccines caused their own reactions, paralysis or polio, brain damage, lockjaw, and more. When doctors had tried to suppress the symptoms of vaccine-induced typhoid with a stronger vaccine, it caused a worse form of typhoid, which they named paratyphoid. But when they concocted an even stronger and more dangerous vaccine to suppress that one, they created an even worse disease, which they didn't even have a name for. They didn't want to tell people what it really was. Their own Frankenstein monster, which they themselves had created with their vaccines and suppressive medicines. They wanted to shift the blame away from their vaccines, so they called it the Spanish influenza, which explains the soldiers. But what about the regular civilian people? Did this maskless body collector know the truth? That the First World War was of a short duration, so the vaccine makers were unable to use up all of their vaccines on the soldiers, so they decided to sell it to the rest of the population. They drummed up the largest vaccination campaign in U.S. history. Their propaganda claimed the soldiers were coming home from countries with all kinds of contagious diseases, and that everyone must have all of the shots on the market. The people believed them. First of all, they wanted to believe their doctors, just like today. And second, the returning soldiers certainly had been sick from their vaccines. So no, a virus or bacteria did not cause the Spanish flu, and vaccines did not eradicate it. Vaccines caused it. And once they were sold out of vaccines, the pandemic instantly stopped. But what is more interesting about Dr. McBean and her doctor parents living through the Spanish flu pandemic is that their testimony, too, debunks the contagion myth. She tells us that the flu hit only the vaccinated. Those who had refused the shots escaped the flu. Her family had refused all the vaccinations, so they all remained well all the time. Hers seemed to be the only family which didn't get the flu. So her doctor parents went from house to house doing what they could to look after the sick, as it was impossible to get a doctor then. Just like the failed contagion experiments we've already discussed, if it were possible for germs, bacteria, virus, or bacilli to cause disease, they had plenty of opportunity to attack her parents when they were spending many hours every day in the sick rooms. But they didn't get the flu, and they didn't bring any germs home to attack the children and cause anything. None of her family had the flu, not even a sniffle, and it was in the winter with deep snow on the ground. Only the vaccinated died. Then we have the strange case of the two Russian conjoined twins, Masha and Dasha, who each had their own lungs and organs, but because their circulatory systems were interconnected, they shared the same blood. Dasha was always prone to colds, but not Masha. And as children, one would become ill with a childhood disease like measles, while the other would not. If the measles was a bug, it would have been in both of their bodies and in their collective bloodstream. So why didn't they both get the measles? This phenomenon was seen over and over again with the girls. Flus, colds, and other childhood diseases were all experienced separately, never together, which would explain why neither Dr. Sellards of Johns Hopkins nor Dr. Hess of New York were able to transmit measles or chickenpox in all of their experiments, because germ theory is nonsense pseudoscience. We'll get into what those really are later. But first, in order to address virology misconceptions and fraud, we need to address polio. After all, didn't Dr. Salk's 1955 polio vaccine eradicate polio like we are all told? Well, only if Dr. Salk also invented a time machine to go back 7 years to December 1948 and give out his vaccines, when polio cases were almost non-existent already, the exact same year when farmers stopped spraying food crops with lead arsenate pesticides when DDT became widely available. Unfortunately, it was short-lived,
As it turned out, DDT was an even more dangerous neurotoxin than lead arsenate, and right after the mass crop spraying of DDT in early 1949, a new spike of polio cases suddenly arose that was 80% higher than the lead arsenate was causing. It didn't help that they were also spraying this neurotoxin on the streets, in the water supply, in their gardens, and even on people and children. The word poliomyelitis was coined from the Greek words polios, meaning gray, and myelos, meaning marrow, plus itis, which means inflammation. Thus the word means inflammation of the gray matter of the spinal cord, the brain, or wherever in the body gray matter is found. Every cell in the human body contains a small amount of gray matter. Inflammation is the result of poisoning. This poisoning damage, decay, and disintegration of cell structure can result in paralysis and, in around 5 to 10% of cases, even death. As Dr. McBean points out, isolated germs have never been known or proven to attack and cause decay and disease in any part of the body. And so polio was never eradicated by Salk's vaccine because vaccines do not eradicate nervous system poisoning. Polio is not a virus. That is an unproven theory. It is a toxic inflammation disease that was simply rebranded or renamed into several other medical terms like Guillain-Barré syndrome, transverse myelitis, multiple sclerosis, and a few others that have the exact same poisoning symptoms of gray matter that polio did, so they can give credit to the vaccine for eradicating the disease and now blame a new disease for the damage their same vaccines are causing. There they are, in Pfizer's 9-page list of 1,300 diseases their COVID-19 vaccine has caused worldwide: Guillain-Barré syndrome, multiple sclerosis relapse, multiple sclerosis relapse prophylaxis, myelitis, and myelitis transverse, or transverse myelitis, or polio. We see this over and over again when sanitation and hygiene improvements, the stopping of certain vaccines and toxic medicines, or the removal of environmental toxins already has a disease on a downward trend toward eradication. And then they come out with a vaccine near the end of the trend and claim it eradicated that disease, as seen here in the US and UK when the Salk polio vaccines came out in the late 1950s. And the same here with measles, scarlet fever, typhoid, whooping cough, and diphtheria. All of them were declining and well on their way to rarity, and then the vaccines came out and took credit for it, like the diphtheria vaccine in 1920, which may have even been the cause for a 2-year spike in diphtheria. Whooping cough was nearly gone when its vaccine came out in the late 1940s, and the 1963 measles vaccine is a joke, as it was less than 1 death per 100,000 people who expressed it since 1947. Dr. McBean's book, The Poison Needle, covered in detail the other causes of polio besides vaccination and pesticides, and you can pause and look at this overview if you like, because we need to move on now and look at virology and virologists and ask what it is they are doing to claim contagious viruses exist and cause disease when 0 real physical experiments in the world have ever been able to prove so. First, we need to ask, what is the cytopathic effect theory of virus particles? What is viral replication theory? Where do these unproven theories even come from? And what is a virus particle? This large area on the bottom left is the corner of a dying, rotting, and decaying green monkey kidney tissue cell in a virologist's petri dish that has been starved of nutrients, poisoned, broken down, and decayed further with antibiotics like gentamicin, amphotericin, phenol red acid dye,
and kidney-dissolving trypsin enzymes for many days, then poisoned further with fixative neurotoxic preservatives like formalin, formaldehyde, glutaraldehyde, and then dehydrated in alcohol baths of ethanol and acetone to drain water from the tissue sample, then put into a mold filled with epoxy resin, glue, and wax, then frozen to harden it, then sliced into micro-thin pieces, then stained with heavy metals made of lead and uranium acetate, and then bombarded with 150 to 300 degrees Celsius heat beams under an electron microscope to show us this picture of a still, non-moving piece of cellular debris, which virologists label a virus particle. If you wanted to study and understand living human biology, would you study living active human beings, or would you dig up and dissect the corpse of a 3,000-year-old decayed mummy and just take a wild guess? To get an idea of just how small this chemically manufactured particle is, the smallest measurement on a ruler is 1 millimeter, and these particles are between 50 to 150 nanometers, or 0.00005 to 0.00015 of 1 millimeter. So what is this particle? It is a theoretical guess based on these kinds of electron microscope pictures that have been put through all of the ludicrous chemical procedures we have just described, and these pictures only. When we see pictures like this, these are just artist drawings and computer-generated pictures of what virologists think that black and white two-dimensional image might really look like. Nobody has ever seen one. It's imagination only. The theory is that it is this: a bunch of genetic RNA and DNA material containing instructions for making new copies of itself inside a host cell, with a viral envelope made from fatty lipid molecules taken from cells in the host, with surface spike proteins that help the virus recognize and bind to cells in the host organism. You can see that too, right? It is not alive. It has no nucleus, no digestive system, no excretory system, no respiratory system, no circulatory system, no endocrine system, just genetic material wrapped in a ball of fat and protein. The cytopathic effect theory states that somehow this dead ball of genes, fat, and protein can find its way inside a person's cells and by some form of rainbow magic cause an enzymatic change in the structure of the host cell, even though it has no enzymes to do so, which causes lysis, or the breakdown of the outer membrane of the cell, which blows the cell membrane open, killing the cell or rendering it unable to produce. The viral replication theory states that people sneeze, breathe, or cough these dead, lifeless balls of genes, fat, and protein through the air, and they somehow make their way into other people's cells, first by attaching to cells via their spike proteins, and then using more unexplained rainbow magic to somehow penetrate the cell membrane, where they use more rainbow magic to somehow get the cell to make copies of them and explode them out of the cell to go do the same to other cells and other people. This piece of candy is not alive. It has no nucleus, no digestive system, no excretory system, no respiratory system, no circulatory system, and no endocrine system. It's just genetic peanut and cocoa plant material wrapped in a ball of fat, sugar, and protein, just like a virus. If someone told you this dead piece of candy could somehow make its own way into your garbage bag, use rainbow magic to multiply into a dozen more pieces of candy, and explode out of your garbage bag back onto your counter, you would probably call them insane. And if they tried to sell you a poisonous spray you could put on your countertop that they claim stop this candy from multiplying and covering your kitchen counter, you would most likely suspect a snake oil salesman. The first glaring problem with viral replication theory is that if it were true that these alleged particles go into the lungs, multiply, and explode out all over the place, then it would be very easy to take a sick person's lung fluid, snot, sputum, prepare an electron microscope slide, and these particles should be found in there. After all, the lungs are a living, breathing tissue culture, or a living virologist petri dish, if you want to think of it that way. But the shocking truth is, these alleged virus particles have never been found in the fluids of human beings, sick or healthy. Virologists are taught in school that it can't be done. The particles are too small, and that's just not the way they do it.
There's not one published paper in the scientific literature finding this thing in any biological fluid of any sick human or animal. Anywhere. And everybody agrees. All the virologists agree. They say that's not how you find a virus. We have 150 institutions, the NIH and the CDC, etc. We asked them, can you show us a paper finding this particle in any fluid of any sick person? And they say we don't have such a paper.
Claim they don't have an isolate, but
they claim they cannot find the virus in any fluid. Now, even though they say there's a reason for that, because that's not how they do it. Now, here's the first question. Since the viral theory is I breathe in this virus, it uses my lungs as a culture, right? And so then it grows, and then it's teeming with viruses that get spewed out in the world. Why can't you find it in your lungs? It's a culture.
Oh, your point is if you do a direct electron microscopy of the fluid, you won't see this.
Never.
Okay, yeah, I see.
Never, not once. We asked a guy who was 20 years, senior virologist at Wuhan Institute of Virology, 20 years head of Yale Virology Laboratory. Can you find SARS-CoV-2 in any fluid of any person you say has COVID? He said no. Why not? Because there's not enough virus to find. Okay, we asked him, what about if you put 10 people with supposed COVID and you mix their bronchial fluid together? Is there enough? No. 100 people? No. 1,000 people? No, not enough to see. 10,000 people, you mix their fluid together. Would you then have enough virus to see? And he said no, and then he wouldn't answer anymore.
But he claims what we see on the EM, on the electron microscopy slides, are in fact virus particles. The little bleb
what
things.
What he says is you have to culture them. There is no virus to see in your lungs,
right,
or in your snot.
Enough that they would admit.
Anywhere.
Yeah.
So then what is a virology culture, and how is it they can see this alleged virus particle after a culture, but not in a sick person's snot? The technique was invented by a man called John Enders in the 1950s. Virologists take a person's snot, mucus, sputum, and put it in a petri dish that has dying, rotting kidney tissue from green monkeys. Then they add bovine calf serum, like cow broth, sometimes milk, and then they starve the fluid in that petri dish of nutrients so that the cells in the tissue start to break down and deteriorate. Then they help it along further by adding kidney-specific nephrotoxins, like the antibiotics gentamicin and amphotericin, and sometimes a kidney tissue-dissolving enzyme called trypsin. Then they wait, and these chemicals break down the cells into millions of tiny particles, and then they do all of the other things we discussed already to prepare it for electron microscopy, and they say, aha, there it is, the virus particle. We can see it now. There are obviously many problems with this procedure. First of all, there are multiple sources of genetic material inside a human's snot or lung sputum, including lung tissue itself, debris from dead human cells being excreted through the lung excretory system, bacteria and microbes living in the lungs, food that went down the wrong pipe, inhaled plant pollen, RNA, and DNA from the air, inhaled bugs, and many more. Then, the monkey kidney tissue itself has its own genetic material, as does the bovine calf serum, as does the milk, so there is no way to tell what genetic material came from what in the petri dish, or what you're even looking at under the electron microscope. It's just a giant soup mix of all sorts of genetic material, so you can't even say where that particle came from. Is that particle from a virus? Is it from the milk, lung tissue, monkey tissue, bacteria, plant pollen, calf broth? Who knows? The second problem is electron microscopes take still pictures, not moving. Here is a man standing outside of his door with keys in his hand. Has he just come home and is going into his door? Or is he just leaving and just locked his door? You have no idea without the rest of the footage, to be sure. So did these particles go into that cell, multiply and explode out of it, or did the fact that the virologist just purposely put a bunch of chemicals in a petri dish that purposely destroy and break down the monkey kidney tissue, milk, bovine serum, human lung tissue, dead cell debris, bacteria, pollen, and other cells in that petri dish, and cause the cells to break down into millions of smaller garbage particles as the cell died, and that's what those particles really are, dead cell debris. Cytopathic effect is just a theory, because no person in history has ever, and I mean ever, observed that particle going into a cell, multiplying, killing that cell, and blowing out into millions of copies of the exact same particle, because that particle is too small to be seen without a still electron microscope photograph, period. Those particles have never been purified and isolated to even check that they are all copies of the exact same thing, ever. No virologist disputes this. In fact, we are all told about autolysis, the breakdown of all or part of a cell or tissue by self-produced enzymes from the dying cell, not viruses. Here is a single-cell organism, just like any of your cells, experiencing regular death, and as we see, it too naturally breaks down into millions of nanoparticles of dead cell debris as it dies and deteriorates. That's what dying cells do. This leads to another glaring problem with virus theory. We are told about apoptosis, the natural programmed death of our cells that leads to the cell's nuclear fragmentation, meaning breaking apart into tiny pieces. Also DNA fragmentation and mRNA decay, which would look something like this. Look familiar? We are told the average adult human loses between 50 and 70 billion cells out of its alleged 37 trillion each and every day. But are we sick every single day from the natural death of 70 billion cells? Of course not. Then where is the proof that a theoretical virus particle allegedly killing a mere fraction of those naturally dying cells causes any illness? There is none. This is not science. This is nonsense. Why on earth would virologists be using chemicals and enzymes like amphotericin, genomycin, and trypsin that cause cytopathic effect breakdown of monkey kidney cells to prove that a virus particle killed the cell and not the chemicals they used that do the exact same thing? It makes zero sense. And why are they using green monkey kidney tissue? Before we can examine and easily understand the errors in laboratory virology studies and procedures, we need to discuss and understand virus isolation and purification. Why is that so vital? Because if you're going to claim that that particle is a unique thing called a virus and not just normal garbage from a deteriorating cell, you have to prove it and then stick it in somebody and make them sick with it, which has never, ever, ever been done in the history of the world, ever. Suppose 3 people ate a chicken soup and all got sick with stomach cramps, vomiting, and diarrhea, and we had a theory it might be from the chicken in the soup. In order to be certain, first we'd have to isolate the chicken chunks only from the rest of the soup by putting it through a strainer to get most of the other soup ingredients and fluid out. Then we'd have to separate the chicken chunks only. That's isolation. Pretty simple. Then we'd have to wash those chicken chunks off with distilled water to make sure they aren't still coated with other liquids and ingredients from the soup. That's purification. These are not. Then we'd simply feed those people this isolated and purified chicken and see if they got sick with the exact same symptoms: stomach cramps, vomiting, and diarrhea. If they did, we have a good suspicion that it is the chicken, but we're still not 100% certain yet. We'd still have to run a control experiment where we feed them the rest of the soup without the chicken and see if they still get sick with stomach cramps, vomiting, and diarrhea. If so, it might be something from the broth or something else in the soup that the chicken chunks soaked up while they were in the soup. Fairly simple. But we're still not done yet. If it was the chicken chunks and not the control soup, we'd then have to run a genetic sequencing on that chicken to make sure it actually is chicken and not some substitute made of tofu, soybeans, corn syrup, and chemicals to make it smell and taste like chicken. So we're absolutely certain in the future that it was chicken that is the problem and not some fake chemical substitute. Now let's say we want to prove that this particle labeled a virus actually is a unique thing called a virus and causes an illness. We'd simply take a sick person's snot or sputum, macerate it in a liquid solvent to break it apart a bit, pour that through a nanoparticle-sized mesh filter, just like a spaghetti strainer, to get rid of most of the larger debris bigger than the particle, so that we only have at least things of that particle size or smaller left in our sample. We put that filtered fluid in a density gradient centrifuge and spin it around until all of the materials in the test tube are in banded layers by weight and density. Just like your lighter socks and underwear stick to the washing machine wall while the heavy jeans and shirts fall in the middle, or just like these materials in this glass are separated by density. Then, we'd take a nanopipette and extract only that density band of material that only has these viral particles in them. And we have to genetically sequence them using either the Sanger method or the Maxim-Gilbert method to get the full genome, or see if that particle even has a full unique genome, to make certain we actually have some kind of unique virus particle and not just broken down partial fragments of normal everyday genetic material from the thousands of other things in the snot or sputum sample. In fact, this entire isolation and purification procedure would be even more important to do if you use the virologist's petri dish culture instead of just taking the snot sputum directly from a sick person, because now you'd have to separate, weed out, and eliminate all of the additional genetic material and whatever else they use to grow the culture. Then we simply inject or spray that isolated and purified virus particle material only into healthy people and see if those particles make them sick with the exact same symptoms as the sick person whose snot or sputum sample we used to get those particles from in the first place. That's isolation and purification. So simple a child could understand it. Now let's ask our virology experts.
What do we mean by virus isolation or virus purification? These are jargon words in virology, and they're not very precise. They mean different things to different people.
Now, Dr. Gallo and Dr. Fauci talked a lot about isolation and purification. Can you tell me what the difference is between the two?
Isolation, what was it?
Isolation and purification.
Of the virus?
Yes.
Well, you isolate a virus by finding the virus which causes a disease. And you purify a virus by making just by purifying it so you get a pure virus.
Okay.
I don't understand what the issue.
They interchanged the two, and I wasn't sure
I see.
If it was the same thing or if it was two totally different.
No, it depends on how they use it.
Okay. Can you explain the process of HIV isolation?
Well, didn't Dr. Gallo do that? I mean, he actually isolated it. So,
Yeah.
I mean, why should I do all of this? This is all textbook stuff you're asking me.
I'm not quite sure what's behind your question about isolation.
I don't want to be your textbook. You know? I got other things to do.
Now, in their defense, it would appear that virologists are taught in their school and training that these virus particles are too small to isolate and purify, and run a full Sanger or Maxim Gilbert genetic sequence to prove that it is a unique thing and not just partial genetic material from regular cellular debris. And that may have possibly been true before the 1970s, but that has not been the case for a long time now. And yet nobody has ever isolated any virus and sequenced an actual genome, let alone make a person or animal sick with the same symptoms by injecting or spraying those particles only with no other poisons, rotting flesh, or contaminants, ever. These so-called Petri dish virus particles that they see under electron microscopes are usually around 50 to 140 nanometers in diameter. Yet, when we look at a bacteriophage, they are anywhere from 28 to 200 nanometers in total height. And yet the capsid, where their genetic material is contained, is only 28 nanometers in diameter, much, much smaller than an entire virus particle. And yet scientists have isolated, purified them, and sequenced a full genome for them. Then we have exosomes, which are packets of protein, messenger RNA, and microRNA that are believed to assemble inside of a cell and then leave the cell to communicate with other cells in the area, among other functions. And exosomes are only 30 to 200 nanometers in diameter, nearly the exact same size as so-called 50 to 140 nanometer virus particles, and as you can see, nearly indistinguishable from alleged virus particles under electron microscopy. And yet, Dr. Andrew Kaufman sent me a paper showing that exosomes too have been isolated from blood plasma, purified through density gradient centrifuge, and even genetically sequenced. Which brings us to the nanotechnology industry and IBM, who has recently unveiled the world's first 2 nanometer chip technology for semiconductors, able to not only manufacture 2 nanometer nanosheets, but also to attach them to nanochips. So let's put this in perspective. Are we really to believe that scientists can isolate, purify, and genetically sequence exosomes the same size as alleged virus particles, also isolate and genetically sequence the capsid of a bacteriophage that is literally the size of just 2 spike proteins on an alleged virus particle, and IBM can manufacture a 2 nanometer sheet 70 times smaller than an entire virus particle and attach it to a nanochip? But virologists can't find this giant particle in any person or animal's lungs or sputum. And even when they manufacture this particle in a petri dish, they can't then isolate, purify, and genetically sequence them? Really? It is painfully obvious that they are discouraged from doing so, because it will prove that these particles are not unique organisms called viruses, but just broken down fragments of dead cellular debris with identical partial genetic material from the broken down cells they come from. The pharmaceutical industry stands to lose tens of trillions of dollars in vaccine, cold and flu remedy profits, and over 20,000 prescription drugs to treat lifelong vaccine-caused allergies and illnesses. If the truth ever gets out to the public that viruses don't exist and the germ theory is a debunked fraud. What fraud? The purposeful creation of fictional, computer-generated in silico virus genomes in order to purposely abuse and misuse PCR tests and antigen or antibody tests to con the public into believing they have somehow caught a fictional contagion that they need medicated or vaccinated against. Whether a person is expressing so-called symptoms of a detox, toxemia, or nutritional depletion, or feels completely healthy, the only reason they believe they have caught a theoretical virus is because either a PCR test or an antigen test has told them so. But very few people even understand what these so-called tests really are, or understand that they can detect no such thing as a hypothetical virus particle. That's not what they do, that's not what they are designed for, or how they even work. So let's use an easy-to-understand analogy for PCR misuse. If we had no idea how tall you were and wanted an exact measurement, we'd use a tape measure that is long enough to measure your entire height in one go. We'll call this first-generation height sequencing. We wouldn't use a regular ruler because it isn't long enough or accurate enough to keep stacking and get an exact measurement, but it is good enough for measuring your feet, shins, knees, thighs, hips, part of your torso, your neck, and your head. And once we knew your exact height with the tape measure, we could closely reproduce that by adding together all of the body part ruler measurements, making a few small errors but getting pretty close. We'll call that next generation height sequencing. Now let's replace height with a full genome. The DNA strand of all living things is allegedly made up of different sequence combinations of only the exact same 4 basic molecules called nucleotides, A, C, G, T. And the RNA of all living things is allegedly made up of A, C, G, and U instead of T. The total number of these 4 basic molecules and the different sequence orders they appear in the DNA or RNA genome determines whether you are a wolf, a sea cucumber, a tree, or a human. Pretty simple. Scientists claim that the full human genome is 6.4 billion base pairs long of sequence combinations of ACGT, and though they've never isolated one and checked, virologists claim that the full genome of an alleged virus particle is around 30,000 base pairs long of different combinations of ACGU for its alleged RNA genome. What PCR does is it takes a molecule that is too small to detect, like G or guanine, in DNA and RNA, and it keeps doubling it until it makes so many copies of it you can finally detect it and see it. If you start with just one guanine molecule and double that 31 times or cycles with PCR, you will now have 2.1 billion guanine molecules that you can now detect and measure. PCR was originally invented by Dr. Kary Mullis for early cancer molecule detection, since tissue biopsies took too long to get results and delayed early detection treatment. The problem is, PCR is only meant to amplify small, specific sequences of DNA or RNA molecules, not entire genomes of 30,000 molecules or 6.4 billion for that matter. It was never supposed to be used for genomic sequencing, and especially not for diagnostic virus detection tests. And here's why. PCR is only made to amplify and detect small, specific primer sequences of already known RNA or DNA sequences out of an entire genome, not the entire genome itself. And just like our tape measure analogy, to find your height for the first time by first-generation height sequencing, if you wanted to find the genome for an alleged brand-new, never-before-seen novel virus like COVID-19, you have to start with first-generation sequencing, since it's a brand-new alleged virus that has never been isolated and mapped. So you would have to use either Sanger or the Maxim-Gilbert method to map the entire genome for the first-time generation sequencing. Let's say you did that with a banana, and then put that full banana genome into your computer database. Then, PCR would be very useful to quickly and cheaply rearrange and put together the smashed fragments of banana DNA or RNA from this banana into the full banana genome using next-generation sequencing, just like our ruler analogy, since you already know the full banana genome to rearrange the fragments into, and you know for certain that this is a banana because you smashed it yourself. The problem is, virologists aren't taught how to do this when they get their silly costumes. Instead of isolating and purifying this particle and running a first-generation genomic sequence with Sanger or Maxim Gilbert, they are taught to create a make-believe fictional in silico genome inside a computer. They simply take the smallest pieces of 50 million broken down genetic petri dish fragments from unpurified fluids from humans, bats, birds, or whatever, mixed with the genetic fragments of bovine calf serum and monkey kidney tissue, all of it starved and broken down by the kidney-dissolving enzymes like trypsin and kidney nephrotoxins like amphotericin and gentamicin, and they put them together like a puzzle to create the genome they want to assign to the fictional virus particle. Then, they design the PCR test to look for common small sequences out of that made-up fictional genome.
They then do PCR tests, which have never been standardized against having found a virus in the first place. So they have no idea whether any of these segments are unique to anything because they've never isolated and pulled out the genome in the first place. And so you could ask, so where do they get the genome? They get the genome because they take some of these segments, they find that they attach to certain pieces in this brew, and then they put those segments into a computer, and it makes what they call an in silico genome. In silico is a fancy word for theoretical, which is essentially a synonym for the word imaginary, which is essentially a synonym for the word make-believe. So, and they standardize this in silico genome against the first SARS virus, which was made in exactly the same way with an in silico, i.e. imaginary genome of an imaginary virus. So then they say, this is now what the new definition of isolation means. We find these pieces of genetic material in this brew that contains, first of all, unpurified snot, and then broken down tissue that contains the exact same kind of sequences and genetic material as does the virus, because it's never been proven that there's anything unique about any of the sequences. In fact, as I pointed out when a friend did a BLAST search, which is a search of the essentially genetic database, he found that one of the primer sequences called the spike protein, it matches 93 human sequences and 91 sequences from other microbes and fungus and other organisms. One of the primer sequences called the spike protein, it matches 93 human sequences and 91 sequences from other microbes and fungus and other organisms. So there's nothing unique about this. They're simply testing for pieces of genetic material, having no idea the origin of this genetic material. So as far as I know, and I know this, I think, as well as anybody could know it, there is not one study out there, there has never been with this virus or many other viruses, that has properly isolated the virus, properly sequenced the virus, and if you can't properly isolate it, you cannot say that a sequence of it comes only from that virus.
And since the alleged virus genome only exists in a computer sequence assembled from normal fragments of genetic material found in everybody's snot, PCR diagnostic tests cannot be used because they are only looking for 2 or 3 tiny sequences of 18 to 300 ACGT combinations long fragments found in the snot of all humans that were used to assemble the fictional in silico genome in the first place. It's like testing a child's wagon for a wheel, and when you find one, claiming that wagon must be an 18-wheeler semi-truck. Now you understand why one of the World Health Organization's primer sequences in their PCR test for SARS-CoV-2 or COVID-19 is found in all human DNA. You didn't test positive for COVID. You tested positive for being a normal human being with normal dead cell debris in your lung mucus, which is one of the normal places where the body eliminates dead cell debris when cleaning itself out. The CDC told us all that COVID-19 or SARS-CoV-2 was a novel or novel coronavirus, meaning brand new, never before known or seen. Yet, in the CDC January 2020 document describing how on earth they came up with a PCR test to detect this brand new, never before known or seen virus, they admit right there on page 42. The real-time RT-PCR diagnostic tests were determined in limit of detection studies, since no quantified virus isolates of the 2019 novel COVID-19 were available for CDC use at the time the test was developed and the study conducted. You read that right. They had no such thing as an isolated SARS-CoV virus to make a test to detect. So how did they do it? Assays, meaning analysis, designed for detection of the 2019 nCoV RNA, were instead tested with known titer RNA copies spiked into a diluted liquid consisting of a suspension of human alveolar A549 cells, which are common used lung cells from a 58-year-old Caucasian male with lung cancer back in 1972. Mixed with a viral transport medium, which is the usual RNA from fetal bovine calf serum, poisoned with the usual gentamicin and amphotericin antibiotics to mimic clinical specimens. So apparently, 50-year-old commonly used lung cancer cells and fetal bovine calf serum are genetically equal to a never-before-known or seen novel coronavirus. 2 + 2 = 5. This is not science. This is fraud. As we touched on briefly, in silico simply means done or produced by using computer software or simulation. Imaginary, make-believe, theoretical, not reality. These are in silico people. Just because they move and look like people is not any proof they are actual real people. No different than the in silico genomes for viruses, like the in silico-based whole genome for SARS-CoV-2 or COVID-19. So what did they do? They went into the NCBI computer database and retrieved an already known non-novel genomic sequence called the Wuhan Seafood Market Pneumonia Virus, or Wuhan Hu1, and then grabbed some other already known corresponding homologous computer genome sequences using the Computer BLAST-N tool. Homologous means similar in structure, though not necessarily in function, as the flippers of a seal and the hands of a human are to each other. So what was it homologous with? Their computer showed their fictional genome is showing an evolutionary relationship with the fictional in silico genome in their database that they named bat coronavirus RATG13, and that both sequences shared a 96.12% sequence match. Now, do you understand where this ridiculous story came from? Human beings share a 99% genomic match with rats and a shared working DNA genomic match of 97.5% with rats and mice, which is why they are used for medical research experiments. Is it really a shock that the boogers of a Wuhan person with pneumonia had a 96% match with the boogers from a flying mouse in a computer database? It is also important to know that because they are using PCR and next-generation sequencing to assemble the 50 million partial fragments of broken-down genetic material in these virology petri dishes into their fake, fictional in silico virus genomes, and because 31 cycles of PCR doubling on a simple 5-molecule fragment like AACCG will now create 4 billion A's, 4 billion C's, and 2 billion G's that aren't really there in real life. When you try to piece these sequences together to create your genome, it gives the computer unlimited artistic liberties to find any sequence it wants anywhere in this petri dish, because the PCR itself creates unlimited molecule sequences that aren't really there. This is called de novo sequences. Imagine if the lotto came out and said, tonight we're going to draw 56 million numbers, and if your 6 numbers come up in a row anywhere in that sequence, you win.
And because you have every possible nucleotide sequence to work with. So you can make whatever you want. Now, once they make one, then they use that as the template to make the other ones, so it looks like that. Because once you make one unicorn and you say people make another unicorn, they make it sort of like the old unicorn.
All right, the SARS-CoV-1 unicorn just happened to be about 30,000 base pairs, so we'll look for something that's about 30,000 base pairs and happens to look like SARS-CoV-1.
Right.
I went back and read the paper. Right. I couldn't believe it.
Yeah, and then that one was made against the bat one in 1996, and that was against the hamster one in 1984, and that was against the bear one in 1906, and that one was made up. So
and even more sinister than that is the way that then became a vaccine. The drug companies then took this in silico genetic sequence from the internet that the Chinese had uploaded from one patient, and boom, they overnight made a vaccine. That's what they admit to. That's what they say. That's what Fauci says. We got this within hours. Once they uploaded the sequence, we took it and made a vaccine.
Right.
Unbelievable. If people knew what they were being, given based on one patient and this uploaded genetic sequence on the computer, it's unbelievable.
Yeah, it's complete make-believe.
Yeah.
It is just flat out. Virology is, because in this study that we did, we actually took this culture, right? So it broke down, and then it breaks down in all these pieces, and then we did an alignment, right? We found the genome.
You did like they did, okay.
Yeah, we did like they did, and we found SARS-CoV-2. But we can also find any other RNA virus as long as you put yeast RNA in there. So you just sprinkle some yeast in there, and then you get HIV and Ebola and measles and SARS-CoV-2, because because it's partly, once you start with all the RNA sequences, but also the PCR process makes de novo sequences. They make sequences which weren't there. The reason you know that is because if you do 10 cycles, you can only get 10% of the genome. If you do 40 cycles, you get 98% of the genome, which means that approximately 80% were made by the process, which is I mean, this is crazy. And yet we all believe it.
That is why the World Health Organization admitted that the high cycle PCR test produced false positives because they instructed all labs to run 35 to 40 cycles, which, as Dr. Cowan just said,
if you do 40 cycles, you get 98% of the genome, which means that approximately 80% were made by the process, which is I mean, this is crazy.
The American biochemist Kary Mullis won the Nobel Prize for inventing the PCR technique.
How do they misuse PCR to estimate all these supposed free viral RNAs that may or may not be there?
I think misuse PCR is not quite. I don't think you can misuse PCR. No, the results, the interpretation of it. See, if you can say, if they could find this virus in you at all, and with PCR, if you do it well, you can find almost anything in anybody. You can find almost anything in anybody. It starts making you believe in the sort of Buddhist notion that everything is contained in everything else, right? I mean, because if you can amplify one single molecule up to something that you can really measure, which PCR can do, then there's just very few molecules that you don't have at least one single one of them in your body, okay? There's just very few molecules that you don't have at least one single one of them in your body, okay? So that could be thought of as a misuse of it, just to claim that it's meaningful. That's what
also,
it is. But it's not. It doesn't tell you that you're sick, and it doesn't tell you that the thing you ended up with really was going to hurt you or anything like that.
If theoretical virus particles really made you sick, then you could not have those virus particles in you and miraculously not be sick. If you have virus particles in you and you are asymptomatic, then those particles do not cause illness, or you would be sick too. End of story. And since no experiment has ever been able to make a person ill by exposing them to another sick person, and since this particle has never been purified and isolated, genetically mapped, has never been directly found in the tissue or snot of any living being, sick or well, injected into another person or animal solely by itself and made them sick, the whole germ theory rests on the baseless claim that this particle somehow enters a cell, multiplies, kills that cell, and goes on to do the same elsewhere, otherwise known as the cytopathic effect theory. That, too, has never been proven or shown to happen. But all that needs to be done to end virus germ theory once and for all is to prove that this particle is just normal dead cell debris from a dying cell and not the cause of that cell's death. And that has actually already been done, not once, but twice. The first time was back in 1954 in a paper called Propagation in Tissue Cultures of Cytopathogenic Agents, meaning toxins that kill cells, from patients with measles, a study done by John F. Enders, who invented this cell culture technique, and Thomas C. Peebles. They actually ran a control experiment where they didn't put any tissue or pus into the culture from a person with measles, or any human being for that matter. They just starved and poisoned the monkey kidney tissue using the same techniques we have already described, and lo and behold, on page 9 of their study, they admitted that a second agent was obtained from an uninoculated, meaning no human measles material added, culture of just monkey kidney cells. The cytopathic changes, meaning destruction of the monkey kidney tissue cells it induced in the unstained preparations, could not be distinguished from the virus isolated from the measles. Just like Dr. Sellards of Johns Hopkins and Dr. Hess of New York could not infect a healthy person with measles by injection from or exposure to a person with measles. In plain English, Enders and Peebles did the cell culture procedure both with the pus from measles patients and did the exact same procedure without the pus of any measles patients, and the monkey kidney cells broke down into identical particles of dead cell debris that could not be distinguished as any different from each other. Enders proved his own procedure of starving and poisoning the culture causes the cytopathic effect, not the theoretical measles virus in the human snot or pus. That should have been the end of virology. But the problem is, Pasteur's personal diary, self-admitting that he was a fraud who lied and cheated all of his experiments, wasn't released until 45 years later. So Enders and Peebles blindly believed that viruses had to be real, without ever first double-checking the premise of that false assumption. As virologist Dr. Stefan Lanka says, it's not that virologists are intentionally trying to deceive the public, but rather that they themselves have been deceived. They simply follow the procedures they were taught and interpret the results in the way they were taught. We can see this clearly in a virology study done by F. Rapp, I. Gordon, and R. F. Baker, who referenced the John Enders measles paper for their own work. They say, monkey kidney cells, however, are unsuitable for investigations of the type reported here, because Enders, Peebles, and Ruckel showed that monkeys and cell cultures derived from monkeys are often infected with an agent serologically indistinguishable from human measles virus, which causes cytopathic changes, meaning cell breakdown of monkey kidney cells in cultures, almost identical with those caused by human measles virus. In other words, instead of realizing it was the starving and poisoning procedure causing the cytopathic cell breakdown in both cultures, they assumed that monkey kidney cells naturally had the same virus as the human measles, and that must be why they couldn't tell them apart from each other. They even go on to say that it seemed likely that the measles virus they assumed was breaking down the monkey cells spreads from cell to cell, both via the surrounding nutrient fluid in the culture and directly from cell to adjacent cell. It is tempting to postulate that the cell to adjacent cell mechanism was mediated in part by the intercellular processes, meaning their own common sense told them that this had to do with the cells themselves and not a virus. But it didn't fit their virus hypothesis, so they stopped thinking about it logically. But what is more crazy is that even by their own logical fallacy, they at least admitted that they shouldn't be using monkey kidney cells for these cultures, as they are unsuitable for investigations of these types. And yet to this day, 55 years later, virologists are still taught to use them, even when their own literature tells them not to. We see this fallacy time and time again, like this paper. Appearances can be deceiving. Viral-like inclusions, not virus particles themselves, but other particles that look exactly like these virus particles, in COVID-19 negative renal, meaning kidney biopsies, by electron microscopy. In vivo means within the living body of a plant, human, or animal, like watching live frog migration or like taking your living kidney tissue out and studying it.
In vitro means a dead artificial environment outside of a living organism, like studying a dead frog, or after your kidney cells are starved, poisoned, and killed, then irradiated and frozen to look at under an electron microscope. In vitro. This paper admits the fraud. Although many of these images were obtained after in vitro infection of cultured cells with SARS-CoV-2 and are thus likely, but certainly not proven to be a true representation of viral particles, we have observed morphologically indistinguishable inclusions, meaning it looks identical in shape under an electron microscope within podocytes, a fancy term for kidney cells, and tubular epithelial cells, both in patients negative from corona disease 19 or COVID-19, as well as in renal kidney biopsies from the pre-COVID era. Did you get that? In other words, this alleged COVID-19 particle isn't unique or novel at all, because they've already been seeing it in kidney biopsies since the 1970s, and they see it in cell cultures now, even in the tissue from people who tested negative for COVID-19. Johns Hopkins warns us about coronavirus, kidney damage caused by COVID-19, and explains how coronavirus might target kidney cells. But yet, they tell us in this paper we looked for SARS-CoV-2 RNA in 8 different kidney biopsies from sick patients who bogus tested positive for COVID-19 and had evidence of kidney disease and were unable to detect virus RNA in renal kidney tissue, despite adequate positive control groups. Later, they discuss viral inclusion bodies, meaning abnormal particles or debris structures which appear inside of the cell in either the cell's nucleus, the cytoplasm fluid inside of the cell, or both of them, which they theorize happens during the course of this alleged virus multiplication process inside of the cell. Yet this paper admits, alternatively, these viral-like inclusions could actually represent microvesicular bodies containing exosomes before their release onto the cell surface. What they are talking about are these pouches inside of a cell, which are called endosomes, which contain the cell's exosomes before they burst out of the cell to go and communicate with other cells. They are admitting they might be mistaking endosomes and exosomes for their theoretical viral-like inclusion particles because they can't tell the difference. People who are ill and self-healing are often proteinuric, creating extra proteins and exosomes in the body to repair damaged tissue, and they say this could lead to the mistaken assumption that these represent virions or virus particles. They admit that individual exosome sizes vary, but they are generally between 30 and 150 nanometers, which fall within the size range reported for SARS coronaviruses. As we stated before, they can see, isolate, and map exosomes, but claim they can't do the same for virus particles because they are too small, despite being the same admitted size of exosomes. And so they get a fictional in silico computer genome because they don't have a genome, because they are just garbage, virus, dead cell debris. They tell us the potential for confusion of coronavirus particles with normal cellular components was already highlighted in a detailed study by the U.S. Center for Disease Control back in 2003 during the alleged SARS outbreak. And even worse, they admit that recognition of this pitfall of virus-like particles actually dates back to the 1970s, when the potential for mistakenly assuming that normal cellular components, such as phagocytic vacuoles, microvesicular bodies, or extracellular breakdown products, could represent viral particles, virus, dead cell debris, was emphasized after a proliferation or multiplying number of many studies claiming to have found ultrastructural virus particles within different types of cancer cells and fluids. In plain English, these aren't anything special and they don't kill cells. This is what the garbage aftermath always looks like. When the average person who does not have the basic knowledge that you now have looks at a paper like this, the isolation and rapid sharing of the 2019 novel coronavirus SARS-CoV-2 from the first patient diagnosed with COVID-19 in Australia, it looks very fancy, scientific, and overwhelmingly complicated, and they assume, well, they must have isolated it. Look at all of those fancy words and pictures. You're now saying, diagnosed how? The normal cough, shortness of breath, fever, chills, muscle aches, vomiting, diarrhea, and loss of taste of sense and smell normal for every flu and thousands of cellular diseases? Diagnosed with a bogus PCR test or antigen test that can't detect any such thing? Who proved these people had any such thing as COVID to begin with, and how? And sure enough, when we look at how they observed the growth and visualization of this alleged SARS-CoV-2 virus, they tell us electron micrographs of the negatively stained fluid showed spherical and pleomorphic virus-like particles that were at least the right size, 90 to 110 nanometers in diameter, that did have some prominent spikes on it, characteristic of coronaviruses, but this did not look at all like COVID-19. Just like this rubber ball doesn't quite look like a spiked dog chewy toy. So following several failures to recover virus particles with the characteristic fringe of surface spike proteins, like the spiky dog chewy toy has, it was found that by adding trypsin to the cell culture medium, an enzyme that dissolves proteins, immediately improved virion morphology. In other words, even though our patient was diagnosed with COVID-19, after several failed attempts, we couldn't find any spiky virus particles that looked like COVID-19, only these round particles with a few spikes, so we added trypsin to dissolve the protein coating, and then it looked like COVID-19. Yeah, well, we were looking for this motorcycle but couldn't find one, only this bicycle, which kind of resembles a motorcycle. So we stuck an engine on it after the fact, and as it turns out, we really did find a motorcycle after all. That is literally the trusted science of virology. And just like they admitted they shouldn't be using monkey kidney tissue in this cell culture procedure, here, in a Journal of Nature article entitled Fetal Calf Serum Contamination Acknowledged in the Literature, where they explain that FBS, or fetal bovine serum associated RNA, meaning the genetic material from the blood of the cow fetus that makes up the serum itself, is co-isolated along with cell culture-derived extracellular RNA and interferes with the downstream RNA analysis. First of all, co-isolated is another virology oxymoron. Isolate means one thing and one thing only. You cannot co-isolate it with something else. That is not isolation. But they explain that the reason it interferes with the downstream RNA analysis is because many evolutionally conserved fetal bovine serum-derived RNA species can be falsely mistaken as human or mouse RNA sequence transcripts. Furthermore, they admit that fetal bovine serum RNA transcripts can themselves be taken up by the other cultured cells and affect the results of highly sensitive gene expression profiling technologies, so precautions for using it in cell cultures are warranted to minimize the interference and misinterpretations caused by fetal bovine serum-derived RNA.
Yeah.
This is nonsense.
Yeah.
Then they say they find the genome, and I can, again, read you 20 papers. Anytime you put fetal calf serum in there that contaminates the specimen, you cannot find the genome. You don't know whether this is coming from the kidneys, from the original unpurified snot, from the fetal calf serum. They put it all in a thing. They put it through like a puzzle maker. It's called alignment.
Yeah.
They create the genome, which never exists in this solution. This frank nonsense.
So all that needs to be done to easily disprove this is to do the control experiments, where you do the cell culture procedure, but without adding any genetic material from a person or animal, sick or healthy. Or, if you wanted to disprove the idea that monkey kidney tissue and cell cultures derived from them must be infected with some virus indistinguishable from human viruses, instead of using monkey kidney tissue, you'd start with a culture of human epithelial cells that are visibly healthy under the microscope, since it is alleged that epithelial cells are often the first place that is attacked by viruses, and run a control experiment with nutrient starving and poisoning versus leaving it alone and giving it nutrients. And see if you can, A, observe these viral-looking particles that are morphologically indistinguishable from the published alleged virus particle. B, observe cytopathic effect and cell destruction despite no possible viral material added to the culture, to prove that it is the procedure itself causing the cytopathic effect. And then C, sequence the genetic material from that culture in the same software and PCR technique virologists use to see if you can also have the computer rearrange those broken down RNA strands of human epithelial and fetal calf serum and make them match one or more of the fictional in silico virus templates in the computer database, despite not having any viral material in it, which has just recently been successfully done by the legendary virologist Dr. Stefan Lanka on a piddly budget of just $42,000. Yet the multi-trillion-dollar medical industry has never bothered to do this simple control experiment even once ever since John Enders. Of course, it has been said that BlackRock and Vanguard own the world. They most certainly own the top two controlling stocks in the pharmaceutical industry, and they own the top two controlling stocks of every major mainstream media outlet. So the BlackRock and Vanguard-controlled big pharma cartel will never let the BlackRock and Vanguard-owned mainstream media inform the general public or the medical industry that Dr. Lanka has debunked viral cytopathic effect theory, as only an idiot would take down their own multi-trillion-dollar pharmaceutical empire. Especially with all of the political power that fake plandemic scares have to bring in the UN Agenda 21 Great Reset. You would never in a million years bow to forced or coerced medical tyranny and vaccine digital ID passports if you knew Dr. Lanka took a virology cell culture with regular industry standard healthy human epithelial cells, and instead of adding any human snot or RNA, he just added simple standardized yeast RNA, then starved that culture, poisoned it with the same antibiotics that virologists use, and the cells broke down in true cytopathic effect with no possible viral material added, and then was able to use their next-generation sequencing and PCR techniques to rearrange the broken-down human epithelial, bovine calf, and yeast RNA fragments into the full in silico genomes of SARS-CoV-2, HIV, Ebola, and measles. Unlike John Enders, Dr. Lanka's control experiments were repeated 3 times in duplicates, because real science should be repeatable. In order to run a proper control experiment, you first have to look at your stock-purchased human epithelial cell lines to make sure they are healthy and showing no cytopathic effect or damage before the experiment. They look like this. Then, in control group 1, those healthy human epithelial cells were put in a culture medium known as CNT07,
which is just 21 amino acids, 19 minerals, 14 vitamins, growth promoters like insulin and hydrocortisone, which stimulate the cell growth, but do not contain cholera toxin or poison phenol red. Then the healthy cells were stressed by adding 1 dose of ABAM. ABAM is simply the antibiotic antimycotic from Thermo Fisher, used by virologists, which is made up of the poisons penicillin, streptomycin, and amphotericin B. It is then left alone for 1 day and then 3 days, and we can see that after 1 day of being poisoned with penicillin, streptomycin, and amphotericin B, there isn't much difference from the healthy cell line. But after 3 days, there is already a noticeable change and cytopathic effect happening from the poisoning. You can see the giant white blotches appearing everywhere. In control group 2, the most important, the same batch of healthy human epithelial cells are given DMEM and Glutamax. DMEM and Glutamax are culture liquid mediums that support cell health and growth with amino acids and vitamins. And control culture 2 is also given 10% FCS, or fetal bovine calf serum blood, which is used by virologists because it also gives the culture the hormones, vitamins, and nutrients to support the tissue life and help it grow, which is why it's also used in stem cell research. The reason control culture 2 is so important is because it shows us there is no possible viral material contamination in the original human epithelial cell stock, because if cytopathic effect theory were true, it would have caused cytopathic effect. But as we see, despite culture 2 also getting 1 dose of the poisonous ABAM, penicillin, streptomycin, and amphotericin B, there was 0 noticeable cytopathic effect breakdown change between 1 day and 3 days, or even 1 day and 5 days, because the nutrient-rich culture allowed the cells to deal with the mild poisoning. In other words, controls 1 and 2 establish the source epithelial cells are healthy and virus particle free, so long as their environment is kept that way, healthy and nourished. Healthy and nourished, starved, and poisoned. Pretty simple. Next, in control culture 3, Dr. Lanka did exactly what virologists do to prove their bogus cytopathic cell-killing effect virus theory, and absolutely chopped off the last flimsy leg virology had left to stand on. Dr. Lanka now took the already control-proven healthy epithelial cells from the same exact stock as controls 1 and 2, but this time, despite giving the culture some nutrition via DMEM, he withheld the Glutamax nutrients. He reduced the fetal bovine calf serum nutrients from 10% to just 1%, and instead of giving it 1 round of triple ABAM poisoning antibiotics, he gave it 3 rounds of poisoning, like virologists do. And lo and behold, oh my God, there it is, cytopathic effect and virus particles after 3 days of nutrient starvation and excessive antibiotic poisoning, despite having no possible virus material in the culture to cause the cytopathic effect breakdown. These starving and chemo-poisoned children aren't dying from viruses. They are being starved and poisoned. The proven healthy source epithelial cells in the 4th control group were starved and poisoned exactly the same way as starvation group 3, but this time RNA or ribonucleic acids from yeast were added to the cell culture. The reason the yeast RNA was used is because when you introduce the lung fluid sputum from a patient in a normal cell culture, it is loaded with nucleic acids. So this yeast RNA, which is rich in nucleic acid molecules, was added to mimic a patient's lung sputum fluid. But yeast-derived RNA is completely neutral and is not pathogenic in any way. But here, you can see that its presence causes an even greater cytopathic effect, despite not being related to any pathogen. Again, these experiments were repeated 3 different times in duplicates. This is not a fluke. Virology is dead. If you're wondering what vaccines are made of and how they make them, they literally take these broken down cell culture soups of someone's boogers or lung sputum, bacteria, bovine calf serum, monkey kidney tissue, or sometimes aborted human fetal kidney tissue, which this AstraZeneca COVID-19 vaccine insert calls genetically modified human embryonic kidney cells, plus all of the poisonous antibiotics used to break that tissue down, and call that toxic soup a live virus vaccine. An inactivated virus vaccine is where they just take some of the proteins out of that same culture, but not the entire culture. This confuses people because their own theory admits that virus particles are not alive. So how can you have a live virus vaccine and a dead, inactivated vaccine? It's not the virus that's alive in your vaccine injection. It's some of the other monkey, fetal, bacterial, and lung tissue cells that aren't dead yet. Both of these are then mixed with poisonous adjuvants like ethyl glycol, which is antifreeze, formaldehyde, acetone, which is fingernail polish remover, aluminum phosphate, mercury-based thimerosal, aluminum, and the disinfectant phenol. Then they inject all of those poisonous chemicals and rotting animal flesh into you and claim it magically makes you immune from catching a fictional virus that doesn't even exist in reality. Outside of the dead cell debris particles they just created in a petri dish and injected into you. And people wonder why they get sick after a flu shot, or why their children have lifelong allergies and medical problems after taking 30 of these injections. And yet, everyone who has vaccine-damaged children or already knows what this film is talking about are labeled as crazy anti-vaxxers, when you'd have to be crazy yourself to inject you and your children with rotting monkey flesh and antifreeze for health. But hang on, I know for a fact that vaccines eradicated polio. The newspapers told me. Time magazine told me. My teachers and parents told me. That is called secondhand information bias. Did you ever bother to check the scientific literature yourself? Or do you religiously practice blind faith in what unqualified people tell you? Anyone who hasn't thoroughly investigated and understood the so-called scientific proof that there are submicroscopic particles called viruses capable of causing various illnesses is simply practicing blind faith. The 1952 polio propaganda tells you don't get chilled. Why would a virus care if you are chilled? Well, Dr. Eleanor McBean told us one cause of polio is interference of any kind that impedes circulation or normal functioning. It also tells you don't get overtired. Why would a virus care if you are tired or not? Dr. McBean also told us polio can be caused by fatigue and all other practices that deplete vital energy, not a virus. Let's look at the proof that there even was a polio virus to be vaccinated against. A 1947 study done by the New York State Health Department, published in 1949, entitled "A Virus Recovered from the Feces or Poo of Poliomyelitis Patients, Pathogenic, meaning causing or capable of causing disease, in suckling mice." Suckling mice are tiny pink newborn undeveloped baby mice that still get their nutrients from their mother's milk. Right out of the gate, we get the first 3 usual problems with virology studies. The study describes the isolation and certain properties of a virus recovered from the feces or poo of children having symptoms similar but not the same symptoms to those of poliomyelitis.
Wouldn't you want the feces from children who have actual polio symptoms and not similar? The agent differs from poliomyelitis virus in its host range, being pathogenic or disease-causing for suckling mice and suckling hamsters, but not for adult mice, adult hamsters, or for rhesus monkeys. Yet both adult humans and children got polio symptoms, but now this virus is also magically age-selective? The third problem is that Koch's third postulate states that the microorganism, when introduced into a new healthy host, should cause the exact same disease symptoms as it allegedly caused in the sick host that the sample came from in the first place. In plain English, if you suspect your grandmother's heart medication actually gave her a heart attack, and you repeatedly gave that same medication and dosage to 100 elderly female monkeys, but none of them had a heart attack, but a few of the monkeys got diarrhea and nausea, you would then go on to look for another cause of her heart attack. You wouldn't say, well, nausea can be a symptom of a looming heart attack, so that proves it was the heart medication. Polio is alleged to be a virus that attacks the central nervous system. Yet right here they admit in the first paragraph, the disease in the experimental animal differs from poliomyelitis, and that the anatomical response is in the striated surface grooves of the muscles rather than the central nervous system. I'm sorry, but that is not polio. The idea was to inject children's poo feces into mice and hamster brains to see if it destroyed their brain tissue, since another study done by Milzer and Bird claimed that destroyed brain tissue facilitates the isolation of poliomyelitis in mice. But as we know, that is not isolation. But after many mice and hamsters had poo injected into their heads without successfully destroying their brain tissue, it was decided to add an additional group of newborn baby suckling mice, because such animals are unusually susceptible. So they gathered the poo and blood from 42 individuals showing polio symptoms, ground up their poo with sand and sterile distilled water, added blood and algae, refrigerated that overnight, and injected it into baby suckling mice. They injected it into their brains and sometimes into their stomach cavities. And then they injected poo into the heads of regular mice, but mixed with destroyed mouse brain tissue from other experiments. But you're washing your hands and wearing a mask to protect you from this. How is your mask going to protect you from somebody injecting the poo from sick people mixed with ground up rotting mice brains directly into your brain and abdomen? That is not contagion. But the joke doesn't stop there. Later, they explained that the use of very young suckling mice necessitated an unscientific evaluation of the animal tests. So baby mice that died during the first and second days following poo and mice brains injected into their heads were excluded from the calculations. Only those that died, were about to die, or paralyzed during the critical period of the test were counted. This is a joke, because the whole point of a control experiment is to see if the injection of poo in brains itself is causing death or paralysis, or the alleged virus. The fact that the baby mice were dying during the first and second days proves it is the injected poo in brains, not any virus. You can't just throw them out of the study. That is nonsense. What is remarkable is that after taking the fecal poo from 10 patients, mixing that poo with rotting mouse brains, adding poisonous sulfadiazine antibiotics, and injecting that into suckling mice and adult hamsters, only 2 samples out of the 10 caused any death or paralysis in suckling mice, but not the adult hamsters. All of the other tests failed. In the next test, they took specimens from 16 more patients, did the same thing, and all 16 failed to cause paralysis or death, as did the 4 that were also tested in suckling baby mice and adult hamsters. In only one instance, weakness of the limbs was noticed in baby suckling mice injected with poo. But like all newborns, suckling mice already have weak limbs anyway. Next, they tried it on young rhesus monkeys, who allegedly share a 93% common DNA with humans, who were injected with 2 fecal poo specimens and also rotting mouse brains. It was given to 20 young monkeys in their nose, injected directly into their brains and also into their stomach abdomens, and no fever or other signs of illness were noted, even though the same suspension paralyzed 9 out of 10 suckling mice between the 5th and 8th days. Then 16 of the same monkeys were given a different sample of poo the exact same way up their nose and injected into their brains and stomach cavity, and no response was seen, even though that sample had caused paralysis of 7 out of 12 suckling mice between the 7th and 11th days. Next, they decided to try it on 5-hour-old newborn baby guinea pigs. 3 guinea pigs, approximately 5 hours old, were injected into their brains with a soup of mouse brain suspension of mice that had had the poo from a young sick boy named KH injected into their brains earlier. No sign of disease was detected. So then they tried 6 more injections of alleged infected mouse brains into their stomach cavities, but failed to produce paralysis or stimulate the production of antibodies. 3 guinea pigs less than 1 day old appeared totally normal after having their brains injected with mouse brains that had been injected with the poo from a young sick boy named TT. Remember these boys, KH and TT. We'll save the best joke of this farce of a study for last. So polio was alleged to be this highly contagious virus that attacks the central nervous system and causes spinal cord impairment, paralysis, respiratory failure, and even death. Yet these virologists took the poo from 42 different individuals with polio symptoms, injected that poo into the brains, stomachs, and noses of newborn and adult mice, newborn and adult hamsters, newborn guinea pigs, and baby rhesus monkeys, along with the ground-up brains from mice that had previously had poo from humans sick with polio symptoms injected into their brains, and didn't even make any of these animals sick. And the only ones who did get paralyzed or died were some, but not all, of the newborn baby suckling mice and newborn baby suckling hamsters. Even the newborn baby guinea pigs were fine, with 0 symptoms of any illness. They say 84 unsuccessful attempts had been made to induce paralysis by injecting poo and mouse brains directly into these animals' brains. The only thing this experiment proves is that injecting human poo and ground-up mouse brains into the brains of newborn, undeveloped suckling mice and hamsters is too much for their tiny, undeveloped bodies to handle. That's not science, that's animal torture. And they even admit it's not science. They say the alleged isolations to date have all been from patients diagnosed as having had aborting or paralytic poliomyelitis, but since no effort has yet been made to recover virus from patients with other diseases or from healthy individuals, no conclusion based on the association seemed warranted, meaning we didn't even bother to run any control experiments. Oh yeah, and by the way, this alleged polio virus that allegedly attacks and destroys the central nervous system, 54 specimens of spinal fluid have been tested in our suckling mice with negative results of polio, and perhaps it should be pointed out that we have not even found lesions in the brain or spinal cords of our mice. And what about TT and KH, our boys whose poo was used in this experiment? TT was a 9-year-old boy who complained of headache, nausea, and pain in his legs in late August. His torso and back muscles became weakened, and he was hospitalized. But a year later, he was able to discard his back brace and was no longer aware of any disability. KH was a 3.5-year-old boy whose illness began at the exact same time as TT's in mid-August and got sore throat and lethargy. 2 days later, the muscle on his left thigh were found to be very weak. He was sent to hospital and treated, and paralysis was not recognizable 8 months later. The third child, known to have been severely paralyzed, lived on the outskirts of their village, by the farms. So where did they find these kids, and why did they all get sick in August? Well, they lived in a small rural Hudson River Valley farming village with a population of 2,300 people. Here is where they lived. And 6 similar illnesses occurred within the village, all during late August and early September, and were diagnosed with polio. Apparently, the polio virus is so smart, it waits until late August to strike and takes the rest of the year off on vacation. The sanitation in the village was certainly satisfactory, but the area was one in which poultry and dairy farming formed the major enterprises, and certainly other crop farming. One large farm was near the home of TT, and the children had not left the village and had no record of exposure to a known case of polio. So how did they get it? The local physicians recalled how other children who had minor complaints during that summer, including headache, nausea, fever, and leg pains too, but they all recovered rapidly without disease. They didn't catch the virus but had all the symptoms of it. Now, knowing that these local farms where these children lived and played near were being sprayed all summer long with lead arsenate and DDT, known central nervous system destroyers and paralyzing toxins, and knowing that underdeveloped children are more prone to toxicity damage than adults, just like the suckling mice and suckling hamsters in the experiment, do you really think it was a polio virus in late August, or have you finally figured it out yet? Because the virologists haven't, they're still doing these same unscientific experiments to this day.
The audience, you could just shout out, what are the symptoms of COVID-19? What? Fever. Cough. Shortness of breath. Good. Fever, cough, and difficulty breathing, right? I mean, I know that a lot of symptoms have been attributed to this. We can't ask mice if they can smell things. But the respiratory symptoms are the main thing, right? This causes a type of pneumonia, right? It causes respiratory distress syndrome, right? So there's got to be pulmonary symptoms, so shortness of breath, cough, sputum, like coughing up stuff, sputum production, something like this, right? And then fever, we all know fever, right? Those are the most common things, okay? They're taking temperatures of people everywhere for this, right? So we'd expect at least fever. So what symptoms did they actually have? Weight loss and slightly bristled fur. Anyone hear news reports of bristled fur in COVID patients? Weight loss also not a symptom of COVID-19 that's reported. It's a very non-specific symptom. So once again, the experiment fails on this count that this is not the same symptoms as the disease in question.
It is important that we look at antigens and antigen tests, as they too are fraudulently being used to claim detection of these fictional viruses, as well as tiny proteins called globulins, which are falsely named antibodies. Ask your doctor how they even work, and you will get this answer. They are just told to give this test to you, and if it is positive, you must have a virus. Your doctors aren't told that this meaningless test has detected COVID-19 in pawpaws, birds, goats, motor engine oil, blueberries, Spanish sewer water, Coca-Cola, papayas, durians, blank tests, apple juice, tap water straight from the sink, or that British school children figured out on TikTok how to use lemon juice to get positive COVID antigen test results to get their class quarantined and sent home from school for 14 days. Or that these tests are as meaningless as aiming a laser thermometer at someone's forehead to detect a virus.
Okay, we're COVID testing the potatoes. Come on. Oh no, it's got the COVID-19. The potatoes got the COVID-19. Oh no, the potatoes got the COVID-19. COVID-19.
Even though there is zero evidence or proof that virus particles exist outside of a petri dish, cause cytopathic effect breakdown of cells, or are somehow magically airborne and contagious, the presence of globulin proteins or antibodies is claimed to be indirect proof of pathogenic viruses, and that a vaccine is somehow giving a person immunity to such fictional particles. An antigen simply means any substance, such as a toxin or enzyme, that when introduced into the body allegedly stimulates the production of an antibody. Antigens include toxins, bacteria, foreign blood cells, and the cells of transplanted organs. A venomous snake bite or the toxic chemical adjuvants in vaccines are both excellent examples of antigens. We read on MIT Medical's website, how does the COVID-19 antigen test work? While other diagnostic tests for COVID-19, like PCR tests, look for genetic material from the virus, the antigen test looks for molecules on the surface of the virus. Now, you're probably saying, hang on, if they claim that virus particle is too small to isolate, purify, and find out what it's even made of, how in the hell would they know what that even tinier spike protein or any other molecule on the surface of it is? In their usual doublethink and doublespeak, MIT tells us antigen tests often fail to identify people who are actually infected, as the Food and Drug Administration acknowledges. While on the one hand they tell us positive results from antigen tests are highly accurate, while on the other hand they tell us, but there is a higher chance of false negatives, so negative results do not rule out infection. Well, which is it? You can't have a highly accurate test with a high chance of inaccurate false negatives. The answer is neither. Both results are completely meaningless. In more of their doublethink, immunologists tell us that antibodies are specific, meaning if you have a virus protein antigen in your body will make a special antibody that only targets that particular virus protein, but not other foreign proteins that are not of interest to it. Yet at the same time, they claim the reason they put antifreeze, formaldehyde, acetone, aluminum phosphate, mercury, and aluminum adjuvants into vaccines is to stimulate your body to create more antibodies to launch a viral defense. But if antibodies are specific, wouldn't they just make antifreeze antibodies or formaldehyde antibodies that ignore virus proteins that are not of interest to it? Why would I even need an antifreeze antibody if the only way I could get antifreeze into my body is to inject it via a vaccine? But antibody scientists now know that when the body is poisoned, holes are torn in the cells by these poisons, and the cells are destroyed. The body's reaction when cells break down is to form sealing protein substances, glue, globulins, or antibodies, which should be called globulins. These antibody globulins immediately expand in acidic environments and cause our blood to clot to wounds, so the wounds can heal. And these antibodies seal our cells when toxins are injected into our bodies. That is most certainly why injecting toxic adjuvants and vaccines definitely causes a rise in the titer, meaning the amount of antibodies that appear afterwards. The problem is, they claim antibodies are there to deal with foreign antigens introduced into the body, What they never tell you is that even when you get a bruise or a blow to a muscle, kidney, or the liver that forms a bruise, there is an increase in antibodies to help fix the damage, despite no foreign substance entering the body. The medically illiterate public are scared into taking vaccines by ridiculous articles like this. Researchers find antibodies in placentas of pregnant women infected with COVID-19. Ooh. The blood of pregnant women is always full of antibody globulins or globulins to seal the placenta. That's what antibodies do, not to protect the baby from fake computer-generated viruses. That is why the blood of pregnant women has to be diluted 40 times to avoid a so-called false positive HIV test. They admit the most common cause of a false positive HIV result is when the test detects antibodies for a different infection or substance. Great test. False HIV positives include lupus, rheumatoid arthritis, Lyme disease, so-called STDs, having recently had a vaccination such as flu or hepatitis B, prior pregnancies, and even taking part in an HIV vaccine study. Of course, all HIV tests are false positives, because the non-existent so-called HIV virus was also just another in silico computer-generated fictional virus, falsely detected by bogus rigged PCR tests and bogus antigen tests looking for no such thing as HIV. Regular autoimmune diseases that had been around forever were now pinned on a virus and renamed as AIDS. And for both those people and the healthy people without autoimmune diseases who tested positive, AIDS was really just the killing and damaging of the body's healthy cells and the cell depletion of the body's bone marrow by poisonous cell-killing cytopathic HIV chemotherapy drugs like AZT, which had been abandoned decades before as a cancer drug, discarded because of its fatal toxicity. Short answer, if you don't want HIV, don't take a bogus test that falsely claims you have it. If you don't want to die of AIDS, don't take toxic antiviral chemotherapy drugs every day that destroy your healthy cells and bone marrow. But back to antibodies. It is well documented that they are not specific. The binding property of these hydrogen sulfide type proteins is nonspecific. One antibody globulin protein can bind to all sorts of things, and you can easily manipulate them in the laboratory by simply changing the acid level or by adding detergents that change the mineral concentration to make them either bind or not bind. As Dr. Lanka points out, whether or not binding takes place depends on the environment and the state of the proteins, whether acidic or basic, oxidized or reduced. Every scientist who has carried out such experiments or studied them knows this, meaning this theory is nonsense. There are dozens of published antibody studies that debunk the theory and claim that vaccines cause the production of either specific or non-specific antibodies, which then somehow play a role in stopping the reoccurrence of a disease by affording immunity and protection from future theoretical virus particles. One such damning study was done by the Medical Research Council's report on diphtheria outbreaks in Gateshead and Dundee, England, published in 1950, which had two glaring problems. It showed that many of the persons actually in hospitals with diphtheria had far more antibodies in their blood than was supposed to be required for complete protection against diphtheria. And yet the nurses and other people in close contact with these alleged diphtheria-infected people who had no sufficient antibodies themselves remained immune and didn't catch diphtheria from the sick ones with the high antibody counts. Antibodies have nothing to do with any immunity, and diphtheria is not contagious. So they researched further and found people who were highly resistant despite having extremely low antibody count, and people who had somehow developed the disease who had high antibody counts, proving that antibodies have nothing to do whatsoever with preventing alleged viral disease or protecting from it. But the nail in the vaccine coffin was the discovery that children who have agammaglobulinemia, a disorder that makes their bodies unable to produce any antibodies, after breaking out with measles or any other zymotic or so-called infectious diseases, nonetheless recover with long-lasting immunity. They expressed the usual symptoms and signs and were subsequently immune, despite no measles antibodies detectable in their serum afterwards. Can you see the problem here? The whole point of a vaccine is the theory that injecting you with petri dish or test tube created dead cell debris virus particles coupled with toxic chemical adjuvants will stimulate your body to create tons of antibodies. So should this virus debris somehow make its way into your body again later in the real world, these antibodies will somehow attach to the spike proteins around the virus particle, which stops it from being able to attach to the cell wall receptors and somehow get inside of it. If the alleged virus still manages to attach, antibodies can still stop it from penetrating into the wall, except when they can't stop it. Then they claim antibodies can still prevent the virus from releasing its genome into the host cell to prevent replication. And not only that, they theorize that antibodies can also signal to other killer cells in the body to come to the virus for destruction and removal. But if antibody theory were true, then how is it that people with very low antibody titer amounts in their body still don't catch these alleged viral illnesses, or that people with very high antibody counts still do express these diseases? And how is it that children with agammaglobulinemia who can't produce any antibodies recover from measles and any other zymotic diseases normally just like everybody else and never get them again? The answer is because there is no virus to catch, remove, or be immune from. Vaccines and antibody theory are more germ theory nonsense. As Dr. Tom Cowan sarcastically puts it, another example is chickenpox. We get chickenpox once and never again due to antibodies, except if we get shingles, which is also chickenpox, but, well, that's different. Big Pharma has a long history of claiming their vaccine eradicated a disease and then renaming those exact same symptoms and visible disease expressions as several other brand new diseases so they can make new vaccines for those. Usually, their vaccines are causing the illness itself. And they're up to their same old monkey business again right now, this time with the new monkeypox virus. And just like with HIV, they're blaming it on gay men again. And of course, they won't explain why monkeypox originally broke out solely in countries that have no monkeys in them. Luckily for all of us, in September of 2019, the U.S. Food and Drug Administration website announced they approved the first live, non-replicating vaccine to prevent both smallpox and monkeypox. And then in February 2021, a patent was announced for a brand new recombinant pox virus-based vaccine to fight SARS-CoV-2, using a mix of broken down genetic material alleged to come from petri dish cultures with alleged SARS-CoV-2, mixed with broken down pus blisters and scabs from horses and monkeys in starved and poisoned petri dish tissue cultures. I bet you can't wait to roll up your sleeve and inject all of that rotting horse blister pus and monkey blister pus into your body. As luck would have it, just 1 month after these two brand new monkeypox vaccines appear out of nowhere, the psychics at the Nuclear Threat Initiative ran a tabletop exercise in March of 2021 on reducing high-consequence biological threats. The fictional exercise scenario portrayed a deadly global pandemic involving an unusual strain of monkeypox virus that first emerged in the fictional nation of Brinia and spread globally over 18 months. By the end of the exercise, the fictional pandemic resulted in more than 3 billion cases and 270 million fatalities worldwide. Remarkably, their fictional monkeypox outbreak happens on May 15th, 2022, the exact same week we are now told there is an actual alleged monkeypox outbreak. What are the odds? I'd say about as slim as when Johns Hopkins, the World Economic Forum, and the Gates Foundation ran the exact same kind of exercise late October 2019, called Event 201, which simulated an outbreak of a novel zoonotic coronavirus transmitted from bats to pigs and eventually person to person, leading to a severe pandemic. The pathogen and the disease it causes were modeled largely on SARS. And then a SARS-2 pandemic allegedly breaks out from bats less than 3 months later. So how could they know there would be a monkeypox outbreak in 2022? Because just like polio, smallpox variola was not caused by a virus or bacteria. It was not contagious, and it was not eradicated by the smallpox vaccine in 1979, as they always claim. Its bogus proof of existence was created in a virology petri dish using the exact same methods we have discussed to create this meaningless dead cell debris particle under an electron microscope. And then the actual skin blistering dis-ease was just given a dozen new names and new fake boogeymen to blame as the new causes. New names like autoimmune blistering disease, one of the 1,300 diseases caused by Pfizer's recent COVID-19 vaccine, and bullous pemphigoid or bullous pemphigus, also caused by Pfizer's COVID-19 vaccine. Monkeypox? No, Scooby, just vaccine-induced bullous pemphigoid. Or if you prefer, adult-onset chickenpox, aka varicella zoster, 8 different forms of which are also caused by Pfizer's COVID-19 vaccine. You might even ask how on earth a varicella zoster virus infection could even possibly be one of the diseases caused by a COVID-19 injection, but I think you already know the answer by now. As Dr. Cowan said earlier about antibodies, another name for this exact same dis-ease is shingles. HealthSite.com shows us this picture and tells us rare monkeypox cases reported in the U.S. for the first time in nearly 20 years, while the Queensland Government Health Agency shows us the exact same disease and picture but claims it's shingles. But did you know the real name for shingles or monkeypox is herpes zoster? And that the FDA reported the Pfizer COVID-19 vaccine causes 11 different forms of it? Luckily, many people are already catching on to this. And it doesn't take Sherlock Holmes to figure out why there is now a monkeypox outbreak after half the world just took injections that can cause it. So why doesn't everybody who is severely poisoned break out with pox disease skin blisters? There are many causes of pox-like disease expressions due to major vitamin deficiencies, deficiencies in the 12 mineral salts that make up the cells and tissue of our bodies, removal of old fibrin during childhood growth spurts, bed bug bites or cooties, medicinal and environmental toxins, as well as collagen deficiency due to a poor diet lacking in collagen-rich foods, or the consumption of too much sugar and refined carbohydrates, and even oxidative stress, which is extremely destructive to our skin. As Dr. Kaufman points out, oxidative stress causes collagen loss or damage, and collagen is the protein glue that binds the thick bottom dermis layer of our skin to the thin top epidermis layer of our skin. And these pox or blisters occur when that collagen is damaged or destroyed and the hole fills up with either white blood cell pus if you are poisoned or lacking in the mineral tissue salts calcium sulfate and silicea, or fills up with clear fluid if not. And oxidative stress that causes these pox blisters triggers toxic epidermal necrolysis. Pharmaceutical drugs are the leading cause of it, and it has also been reported to occur following measles, mumps, rubella vaccinations. By no surprise, toxic epidermal necrolysis is also one of the diseases caused by the Pfizer COVID-19 vaccine. Another name for that is Stevens-Johnson syndrome, a more severe variant. And what a coincidence, Stevens-Johnson syndrome is also caused by the Pfizer COVID-19 vaccination. Big Pharma actually did humanity an enormous favor by trying to pin this vaccine damage on monkeypox, because it forced us to go back and look at the original monkeypox isolation experiment from 1959, which offers 2 more nails in the germ theory coffin. Their institute got a few dozen completely healthy macaque monkeys in to carry out scientific experiments on, and after 2 months, 6 of them suddenly broke out with pox eruptions, which they suspected was some kind of monkey virus. Sherlock Holmes said the world is full of obvious things which nobody by any chance ever observes, like, what kind of experiments were they doing on these monkeys, and what kind of diets and inoculations were they giving them? If it was a virus, how did they catch it if they all arrived totally healthy and had been in quarantine together for 2 months? Why did it take 60 days to show pox marks when viruses are alleged to cause symptoms within 2 to 14 days? Why did only 6 monkeys break out and not most or all of them if they were exposed to each other for 2 months? Why didn't any humans working with these monkeys at the institute catch it, if monkeypox can allegedly be transmitted to humans? The experiments are littered with the usual suspects: collect the pus fluid from the monkeypox bumps with a cotton swab, poison it with penicillin and streptomycin, just like amphotericin and gentamicin in modern days, stick that on cell cultures of rotting monkey kidney tissue, human amniotic tissue from a placenta, and even HeLa cancer cells from a woman who died 8 years earlier, because we all know how healthy cancer cells are, and let that break down. As usual, to even be able to view this carnage under an electron microscope, they added the tissue-dissolving enzyme trypsin for 4 hours to cause even more cytopathic effect. Then they heated that to 70 degrees Celsius, or 160 degrees Fahrenheit. Imagine what would happen to your cells if you ran a fever that high. You would be dead within an hour. And then they stained it for shadow casting with platinum, which just happens to create nanobubbles on the slides that are visually indistinguishable from most so-called virus particles. But that's none of my business. But what is our business is this: the results of the cytopathic effect breakdown of these tissue cultures. Are you scared of monkeypox because of these pictures? Of course not, because we have all been programmed through constant fictional CGI-generated artistic images that allege deadly viruses are supposed to look like this, which look nothing like the almost rectangular-looking cytopathic effect breakdown in the monkeypox experiments. The paper itself says morphologically, meaning in shape appearance, the monkeypox agent was found to have the size and brick-shaped appearance typical for members of the pox virus group. Virologists tell us monkeypox virus is rectangular in shape. Yet, as if they're too stupid to tell a rectangle from a circle, the BlackRock and Vanguard big pharma-owned mainstream media has been bombarding the entire world with this image, because it looks more like the CGI artistic drawings of alleged viruses that we have been programmed to fear. But they don't even tell you which of these 2 particles you are supposed to fear, the dark oval ones or the light circle ones. They are completely different from each other. Neither one of these are brick-shaped rectangular particles, And as Dr. Kaufman discovered, this image you keep being shown was taken from a clinical sample associated with a 2003 alleged American prairie dog outbreak. But we don't even have monkeys in the U.S. Midwest, so how did prairie dogs allegedly catch it and pass it on to humans? More absurdity. They also explain that the particles on the left were mature, oval-shaped particles, not brick-shaped, and on the right were the crescent and spherical, not brick-shaped particles of immature virions. These viruses are so smart they're just like us. The grown-ups all sit at the grown-up table together on the left, and the kids sit at the kids' table together on the right, never mixing. As the Irish Independent joked, what monkeypox would probably look like under a microscope if they could isolate it. As usual, they scarified or scraped holes into rabbit skin and even injected their toxic cell culture brew into the skin veins of rabbits, but no lesions were observed in their organs. When they injected rabbits with that toxic cell culture brew, it produced only skin lesions at the site where they scraped their skin and rubbed toxins all over it, and also keratitis, which is inflammation of the eye cornea. And when they injected that toxic brew into the brains of mice, it produced encephalitis, or swelling of the brain. But no pox all over the body. Once again, failing Koch's third postulate, that the injection must cause the same skin pox dis-ease symptoms as the original monkeys it was taken from. But the biggest gift this monkeypox paper has given us is that they actually ran 2 control experiments on healthy monkeys, like the John Enders measles experiment and the recent controls run by Dr. Stefan Lanka, where they admit they found the presence of monkeypox virus in the tissue cultures prepared from the kidneys of apparently healthy monkeys. They tell us the first time they had to kill some of the healthy monkeys for other scientific purposes, they took the kidneys from those healthy monkeys, broke that healthy kidney tissue down with trypsin, and cytopathic effect changes developed within 7 to 12 days later, where they found particles identical with monkeypox virus in the healthy cultures. Then they did it a second time from the kidneys of apparently healthy monkeys, and once again, monkeypox-looking virus particles were present in some of the cell cultures, and they did this at a time when clinical illness or visible skin pox was not present in the monkey colony at all. Once again, it took 7 to 10 days for the cytopathic effect to happen in the cell cultures, proving again what we have been saying. It is their procedures themselves to make cell tissue cultures and prepare them for viewing under electron microscopy that are causing the cytopathic effect breakdown of cells, not viruses. Virus particles are dead cell debris produced by their procedures. Pathogenic viruses do not exist, period. The world's biggest vaccine peddler doesn't seem very concerned about wearing a mask, for good reason. He already knows. And it is important that we get the word out, because our economies and societies have been decimated by lockdowns in the name of virus mania. Police are brutalizing people in the name of virus mania, even going so far as to wrap them in bubble tape and haul them away. And families are being torn apart over what amounts to germophobia mental illness out of sheer ignorance driven by non-stop fear-based propaganda. And it must stop. Hopefully you're asking, then what about alleged contagious diseases allegedly caused by bacteria instead of viruses, like sexually transmitted diseases and tuberculosis? They too have been misunderstood and debunked. All germ theory is nonsense, not just viruses, and we have covered that fully in part 2 of this film, Germs Debunk Corona, which, like the Star Wars series, was released before part 1. That film also covers the many sciences and phenomenon of bioresonance, German new medicine, epigenetics and the belief that someone will make you ill making you ill, morphogenetic field interactions, placebo, nocebo, pheromone triggers, living water communication between beings, shared local environmental threats and toxins, and the many other reasons why certain natural healing detox cycles that we falsely mistake for contagious germ diseases can sometimes, but not always, trigger some but not all people in proximity with that person to also start that exact same healing detoxification cycle themselves. No different than women's menstrual cycles synchronizing together, or like chickenpox parties, where nobody ever bothers to ask the really important question: why did only 3 or 4 kids get the chickenpox, but the other 5 or 6 didn't? Or the flu, where Dad and I got sick together for a week, but Sister and Mom walked around the same house taking care of us in constant close contact, but they never got sick. It's because, as you have just seen, it's not from viruses, because viruses don't exist. Period.